as I mentioned, we have two presentations today. One, Andreas Hockhaus will be presenting the clinical highlights of the EHA sessions, and Professor Tomasz Stokłosa will be presenting the biological highlights. So if we start with the biological highlights, and as we begin, I will stop sharing my slides and Professor Stokłosa can bring his up, and I will give a short introduction. So Professor Tomasz Stokłosa is the head of the Department of Tumor Biology and Genetics at the Medical University of Warsaw, Poland. Professor Stokłosa is also a specialist in the field of laboratory medical genetics, and from 2023 is leading the Laboratory of Genetics affiliated at the University Clinical Center of the MUW. He is leading research projects in hemato-oncology and oncology focused on the molecular pathogenesis of hematological malignancies with special interest in CML and high throughput genetic screening, employing next generation sequencing tools. His publication track record includes more than 100 publications in the leading journals with more than 3,000 citations. So a very warm welcome, Tomasz, and please go ahead with your presentations of the CML biology highlights of EHA.
Thank you very much, Nicola. Good morning, good afternoon, good evening, wherever you are. So I'm really honored, and I have to share my personal view because I'm following International CML Foundation starting when I was a postdoc, and I really appreciate your work. And I'm reading all the updates prepared by Tim regularly. I do appreciate it, and I'm definitely not an eminent expert, and it's my debut. So I hope you will forgive me if I will not satisfy everyone with my overview of CML biology. But what I have to share with you at first is my personal, really very, very good impression with European Hematology Association Congress. For some personal reasons, I haven't been attending EHA for the last 5 years. And I was really impressed how it grew, how it is now like not a secondary event to ASH as it was used to be considered 20, 15 years ago. Of course, it's a little bit smaller, but I think the scientific level is really, really top of the notch. And also what I do appreciate, this is this, how you call it, every cloud has its silver lining. So I think the COVID gave us now this chance that we have this hybrid option. So anyone who was registered can still come back and see the lectures, see the presentations, which we usually miss running from one session to the other. So that's my personal impression, but now to the point, to the CML biology, which is my topic. The reason is I was honored to be a chair of the oral session, which was Saturday late afternoon, but still very good frequency. But I will start with a scientific working group session chaired by Nick Cross and 3 excellent presentations. The last, but Jane Apperly will be, let's say, will be discussed in details by Professor Andreas in his second presentation because it's about updated ELN guidelines. But I would like to share with you some of the most important thoughts from the first 2 speakers. The first speaker was Rick van Etten, very well-known expert in CML biology, in CML stem cells. And he has started with what we actually are hearing for the last couple of years. So what are 2 major challenges in CML, or unmet clinical needs, as you prefer? While this is clinical, of course, biology is trying to answer the clinical needs. So the first is the primary TKI resistance, of course, in contrast to secondary resistance, which is caused most frequently by BCR-ABL1 mutations. The primary TKI resistance is still a phenomenon with a largely unknown mechanism, with several hypotheses, but the real mechanism why some patients are not responding upfront is not really clear. So this is, of course, the obvious importance of this question is that we have this need to prospectively identify patients at risk and maybe start with a more potent inhibitor or maybe with some even new drugs which are now starting to enter the market. And then, of course, the second thing, why CML is paving the way for not only hemato-oncology, but also for the whole oncology, is that in CML, we can stop the treatment. However, even after almost 20 years after the initial trials by Francois Mahon, we don't know why approximately half of the patients will relapse and will need back the treatment. So the question here of course, how to identify such patients and how, if we have any tools with which we can increase the proportion of such patients. So Rick presented his work and also work by others on transgenic mice, the famous model developed by Stefan Koschmider with inducible BCR-ABL and inducible disease mimicking chronic myeloid leukemia in mice. And he wrote from his lecture several interesting conclusions. First of all, this is something which maybe is obvious for majority of you, is that the leukemic stem cell clone has selective advantage. But the second conclusion is, I think, worth to note that the compartment of this leukemic hematopoietic stem cell is important not only with respect to the size, but also with respect to the self-renewal capacity. And this may influence the initial response to tyrosine kinase inhibitor therapy. Then one of the other conclusions which also attracted my attention is that the threshold site is definitely required for CML development. This may also has something to do with the treatment-free remission status, if it's successful or not. And what is, for me, as someone who is involved in genetic studies in CML, that Rick proposed that actually it's not only, or it's not even the major reason, additional mutational process implicated. So the data which were presented are showing that CHIP mutation, the clonal hematopoiesis mutation, mutations in this well-known genes such as DMT3A, TET2, they don't need to be present in CML stem cells to affect TKI response. So I would say that this is one of the important things to be resolved, because actually there are other groups and other teams which are showing that those additional mutations have significant role. So if not additional mutations, what can be responsible for these differences in response to TKI or for TIT inflammation? Probably microenvironment, probably epigenetics, maybe both. And then I would like to present you just a glimpse of the second talk by Göran Karlsson from Lund, who was analyzing CML at the single cell level. I skipped this famous and well-known slide about the fruits in the blender, and when you mix them and you do the smoothie, because this is a well-known comparison that when we analyze bulk cell compartment, then it's kind of like looking at smoothie and not seeing a separate kiwi or banana. So it was a very nice lecture. I also recommend who has a chance to listen to this whole talk. He gave a nice overview of the technologies and strategies, what we can achieve by sequencing single cells separated from a bulk population. What is for me the most, I think, important information from this lecture, that actually you may be able even to predict blast crisis at the diagnosis by analyzing some gene expression profiles in a single cell. So what are the conclusions? First of all, you can say which stem cells are insensitive, which are sensitive, and then you can predict a treatment response. The third lecture by Jenna Pillay, as I told you already, will be discussed thoroughly by Professor Andreas in a few minutes. So, as I mentioned, I had this honor to chair the oral session, and I would like to just give you a, how to say, the overview of the 5 oral presentations which were selected by the reviewing committee for this session. The first presentation, we had the pleasure to chair this session with Dr. Ilaria Pagani. I will refer to her because she was also a speaker in a minute. But first presentation by Stein-Erik Gullaksen was exactly trying to answer, can we predict at the single cell level if we are able to tell who will respond to TKI? So this is a very interesting study because it is using mass cytometry. Mass cytometry is kind of a very sophisticated, how to say, approach mimicking flow cytometry, but as long as in flow cytometry you are able to analyze 7, maximum 8, 9 antigens, in this technology, you can actually analyze up to 60 even different antigens on a single cell, barcoding with some metal conjugated antibodies. So, of course, this is not a typical equipment of a diagnostic lab, but hopefully when this technology will become more available, this can really help us, because on some samples from Nordic CML group and also from the ENIS trial, Dr. Gullaksen showed that single cell deep immune and single profile has predictive value. So by analyzing the cells just before the first dose of tyrosine kinase inhibitor and a few hours later, you can actually predict if this patient will be responding to TKI. And it was in the response to dasatinib and nilotinib was actually analyzed. So maybe this is the future. We will have this machine and we will have a patient sample before first pill is given. Then we will analyze it a few hours later and we can predict if this patient will go successfully on imatinib or maybe nilotinib or maybe dasatinib or maybe any other, maybe stronger inhibitor is here necessary. And then the second presentation was by Dr. Ilaria Pagani. Dr. Ilaria Pagani is from Adelaide, from the Harmony Center, but actually Milan is her hometown, so I think it was also a great pleasure, I'm sure, for her to present her data related to the impact of mitochondrial DNA mutations. So, of course, work by Sue Branford team group and also by Dennis Kim showed a genomic landscape of CML, but mitochondrial DNA was rather, I would say, ignored until recent years. So this is a first such a nice attempt to analyze the impact of mitochondrial DNA mutation with respect to the response to tyrosine kinase inhibitors. And what even Ilaria admitted, Dr. Pagani, that actually more mutations were in samples from patients who were TKI sensitive, but the general profile of this mutation was different in TKI responders and non-responders. So I hope that this work will soon be published. And just for the sake of time, I focused on the first two presentations, but I would like to give you at least few words about the last three presentations. So Dr. Zhang from Beijing presented data on overambatanib and ponatinib treated patients, third generation TKI. Interestingly, this first one, overambatanib, I don't want to misspell it, is not available in Europe. It's available in China, I believe, maybe few other countries in Asia. So a typical genomic and transcriptomic analysis of more than 200 patients and actually gene expression profiling. So transcriptomic was more predicting than just the mutations at the DNA level. Dr. Manuela Mancini from Bologna presented a continuous work on SETD2 deficiency. SETD2, a well-known tumor suppressor, which is frequently lost during the progression to CML blast crisis. So very interesting finding that restoring SETD2 expression by proteasome inhibition may help to overcome resistance. So also a very promising abstract. And at the end, Dr. Rinaldetti presented an analysis of selected patients from the famous EURO-SKI trial, so stop kinase inhibitor trial, approximately 200 patients, just to try to answer this question: how can we predict who will be able to achieve TFR and continue it, and who will need to come back to the therapy. So this was an interesting presentation, but finally, the answer is still out there. So still we don't know. So it's still a lot of work to be done. So I have to apologize because there was also one very interesting session preceding this oral session. It was a hematology in focus session about the mutations in CML. It was chaired by Dr. Rzeczkova and also our patient advocate, Mr. Göran Karlsson, and Simona Soverini was presenting the landscape of mutations as she was introduced, the queen of mutations, and then Dr. Hugh Lavalat was also presenting interesting data. So if you have a chance to watch this as a EHA webcast, I strongly recommend. And I would like to thank you for your attention. And of course, I will be waiting after Professor Andreas' lecture for your questions. Thank you very much.